Welcome back to MedStudious. Today we are opening our histological field guide to explore one of the most fascinating structures in the human body, the neural ganglia. When we study the nervous system, it's easy to focus entirely on the massive central processor, the brain and spinal cord. But the peripheral nervous system is far more than just a set of dumb transmission cables. It contains highly organized, localized processing centers that operate like mini brains distributed throughout your body. These are the ganglia. In this comprehensive guide, we will break down the anatomical architecture of these peripheral neural hubs, examine the specific cellular operators that keep them functioning, like neuron somas and satellite glial cells. And finally, we will apply this structural knowledge to devastating clinical pathologies. By the end of this video, you will understand exactly how the localized destruction of these microscopic switchboards leads to catastrophic, life-threatening gastrointestinal conditions like Achalasia and Hirschsprung's disease. Let's begin our journey.
To truly understand the function of a ganglion, we must look at its definition. Medically, a ganglion is a highly structured collection or bundle of afferent or efferent neuron cell bodies, also known as somas, that are located specifically outside the central nervous system. Let's use a visual metaphor to grasp their importance: the neural switchboard. Imagine an old-fashioned local telephone switchboard. Instead of routing every single localized phone call back to a massive, distant central headquarters, which would be slow and inefficient, the local switchboard coordinates calls directly within the neighborhood. Neural ganglia operate on this exact same principle. They act as localized processing hubs that coordinate complex signals between distant areas of an organ directly. A perfect example is your digestive tract. The ganglia of the enteric nervous system locally coordinate the rhythmic, complex waves of intestinal peristalsis without needing to constantly route every single reflexive command all the way back to the brain.
Inside these ganglia, we find the primary operators, the neurons. However, they come in 4 distinct architectural variations. First, the unipolar neuron. This features a single axon protruding from the soma. It is incredibly scarce, found almost exclusively in the cerebellum, where it fires intrinsically to amplify vestibular signals for balance. Second, the bipolar neuron, featuring 2 separate axons extending from the soma. This is a common structure, famously localized in the sensory retina bundle. Third, the pseudo-unipolar neuron. This fascinating crossover type has 1 fused axon leaving the soma that immediately splits into 2 distinct long fibers. You will heavily find these packed into bundles within your sensory and parasympathetic ganglia. Finally, the multipolar neuron. This is the classic primary motor neuron, featuring a single axon but multiple highly branched dendrites. These complex dendritic trees capture thousands of incoming signals from multiple synapses simultaneously, increasing the overall signal amplitude required to actively stimulate and contract your muscles.
So, where exactly does the body deploy these localized switchboards? Let's map the major peripheral neural hubs. Broadly, we divide them into sensory ganglia and autonomic ganglia. Sensory ganglia are the body's data-gathering hubs. The most prominent examples are the dorsal root ganglia, situated directly alongside the spinal cord. Histologically, these are packed with those highly specialized pseudo-unipolar and bipolar neurons, strictly relaying afferent sensory information like touch, temperature, and pain from the body back to the central nervous system. On the other side, we have the autonomic ganglia, which handle involuntary, unconscious, visceral motor functions. This network includes the sympathetic chain running parallel to the spine, as well as massive, critically important regional hubs deployed deep within the abdominal cavity, such as the celiac ganglion, the superior mesenteric ganglion, and the inferior mesenteric ganglion, which collectively regulate the massive blood flow and secretory functions of your digestive organs.
Within the autonomic system, the parasympathetic division follows a very specific anatomical rule. Its ganglia are generally situated directly within or extremely close to the actual target tissue they innervate, resulting in very long preganglionic fibers and very short postganglionic fibers. However, in medicine, there are always high-yield exceptions. The efferent motor neurons of three specific cranial nerves completely break this rule, featuring distinct externally located ganglia far from their target tissues. Cranial nerve 3, the oculomotor nerve, sends its parasympathetic signals to constrict the pupil via the distinct external ciliary ganglion. Cranial nerve 7, the facial nerve, travels through complex pathways to synapse at the external sphenopalatine and submaxillary ganglia to control tears and saliva. Finally, cranial nerve 9, the glossopharyngeal nerve, shoots its parasympathetic fibers directly into the external otic ganglion to regulate the massive parotid salivary gland. Memorize these three exceptions for your anatomy exams.
As these nerve fibers leave the ganglia, they must travel safely through the body. To survive physical stress, they are packaged in a robust, three-layer connective tissue architecture. First is the epineurium. This is the thick, tough outermost sheath enclosing the entire nerve bundle, visually forming the pale, well-defined outer boundary on a histological slide. Next is the perineurium, the middle layer that envelops smaller groups of nerve fibers into distinct bundled cables called fascicles. This layer is crucial because it maintains the vital blood-nerve barrier. Finally, we have the endoneurium, the delicate innermost matrix that surrounds every single individual nerve fiber, intimately wrapping around the axon and its insulating Schwann cell. It is important to remember that a ganglion is essentially a massive swelling along this nerve cable. The tough epineurium and perineurium actually expand and continue directly over the ganglion to physically protect the delicate cell bodies housed inside.
Let's look at a stunning example of how a ganglion integrates directly into living tissue, the enteric or Auerbach's plexus. Remember the rule that parasympathetic ganglia embed directly within their target organs? In the small intestine, this localized switchboard is integrated right into the gut wall. The enteric plexus sits perfectly sandwiched between the inner circular and outer longitudinal layers of the muscularis propria. Its localized job is to coordinate the rhythmic muscular contractions of peristalsis. When you examine this on a histological slide, it is highly distinct. The surrounding smooth muscle fibers will feature long, stretched, elongated nuclei. But nestled between them, the plexus appears as an island of pale, specialized nerve tissue, tightly bound by its own continuous epineurium layer. And as a histological marker, remember that all the massive ganglion cell bodies you see clustered within this specific intestinal plexus belong exclusively to the parasympathetic nervous system.
Let's zoom in to 400x magnification and deconstruct the microarchitecture of an individual neuron housed inside this ganglion. The main body is the soma, the metabolic factory of the cell. Right in its center is a large, pale nucleus, which safely houses the DNA blueprint. Inside that nucleus is a dark, prominent dot called the nucleolus, which acts as a dense factory, continuously producing ribosomes and rRNA. Out in the surrounding cytoplasm, we find the Nissl bodies. These are massive collections of rough endoplasmic reticulum. On a slide stained with a basophilic dye, they appear as dark, highly reactive granules, proving the cell is heavily engaged in continuous protein synthesis. Finally, we have the neurofibrils. These are the microscopic structural scaffolding proteins that act as a biological skeleton, extending from the cell body all the way down the entire length of the long axon to maintain its physical shape.
These highly active ganglion neurons cannot survive alone. They require dedicated cellular support. Enter the satellite glial cells. These are the vital operators of the peripheral switchboard. Think of them as a dedicated pit crew. They physically wrap around the large neuron somas, forming a continuous, protective single-cell sheath. Their primary function is to strictly control the precise chemical microenvironment around the neuron, providing metabolic support and electrical insulation. However, a highly active metabolism leaves a trace. Let's look at the aging marker, lipofuscin granules. As the neuron ages, it accumulates lipofuscin, a yellow-brown pigment that is essentially a harmless cellular waste byproduct of high lysosomal turnover. While a small amount is a normal sign of an aging cell, an abnormally massive, widespread accumulation of these granules in a young patient is highly pathological, indicating a rare, degenerative lysosomal storage disease known as lipofuscinosis.
As a medical student, you must connect this microscopic histology to macroscopic patient symptoms. Let's discuss clinical synthesis, system failure at the switchboard. What happens when these vital, localized ganglia are either congenitally absent or destroyed by disease? The switchboard fails. In the gastrointestinal tract, the absence of local parasympathetic innervation is catastrophic. Without the neural signals commanding the tissue to relax, the local smooth muscle remains in a state of permanent, tight contraction. It permanently loses its ability to dilate and let food or waste pass. This results in a profound mechanical and functional obstruction of the organ. This singular, specific microscopic mechanism, the localized death or absence of the ganglionic switchboard, is the direct driving force behind two distinct life-threatening gastrointestinal pathologies that you will frequently encounter on the medical wards. Let's examine them now.
Our first ganglionic pathology is achalasia. This is an acquired condition driven by the localized autoimmune destruction of the enteric plexus, specifically within the lower esophageal wall. Because those local ganglia are destroyed, the lower esophageal sphincter completely loses its ability to relax when the patient swallows. The primary clinical symptom is severe dysphagia. Patients will often report that swallowing solid food is noticeably more difficult and painful than swallowing liquids, as the food physically backs up. Because unswallowed food pools in the dilated esophagus above the stricture, these patients face a severe risk of lethal pulmonary aspiration. Diagnostically, a barium swallow X-ray will reveal a classic bird's beak appearance, where the esophagus tapers to a severe point at the destroyed ganglion site. Standard medical treatment relies on Heller's myotomy, surgically cutting the unyielding sphincter muscle, along with aggressive balloon dilation or targeted Botox injections to force the tissue open.
Our second pathology occurs at the opposite end of the GI tract, Hirschsprung's disease. Unlike Achalasia, which is acquired later in life, Hirschsprung's is a congenital, developmental condition recognized in infancy. During fetal development, the neural crest cells completely fail to migrate, meaning the enteric plexus entirely fails to form in the distal colon and rectum. Because this tissue is completely aganglionic, the complete absence of parasympathetic innervation prevents the smooth muscle from ever relaxing, severely impairing the localized coordination of peristalsis. The progression is terrifying. This functional obstruction traps feces and gas, causing the healthy bowel above the blockage to massively dilate into what we call a megacolon. If left untreated, the immense internal pressure will eventually cause a fatal bowel perforation. The only definitive clinical treatment for Hirschsprung's disease is the aggressive surgical resection of the entire aganglionic bowel segment, followed by surgical reconstruction.
Let's review a high-yield diagnostic comparison of these two aganglionic obstructions for your board exams. First, consider the affected region. Achalasia strictly targets the lower esophageal sphincter, while Hirschsprung's targets the distal colon and anus. Second, compare the primary etiology. Achalasia is generally an acquired autoimmune destruction of the ganglia that occurs in adults, whereas Hirschsprung's is a developmental aganglionosis present at birth. Third, evaluate the primary clinical risk. In achalasia, the immediate danger is the deadly aspiration of refluxed, unswallowed esophageal contents into the lungs. In Hirschsprung's, the immediate danger is the development of a toxic megacolon, leading to a fatal intestinal perforation. Finally, look at the treatment modality.
achalasia is managed via Heller's myotomy, targeted Botox, or balloon dilation to force the sphincter open. Hirschsprung's disease requires definitive surgical bowel resection and reconstruction to physically remove the dead aganglionic zone entirely.
To wrap up our lesson, let's review the histological blueprint and key takeaways for neural ganglia. First, the hardware. Ganglia are localized functional switchboards situated outside the CNS, composed of specific neuron somas, unipolar, bipolar, pseudo-unipolar, or multipolar, all intimately wrapped in continuous sheaths of supportive satellite glial cells. Second, the pathways. Remember that sensory ganglia generally reside dorsally near the spine. Sympathetic ganglia form the trunk and periphery, while parasympathetic ganglia safely embed directly within their target tissues, with the notable cranial nerve exceptions of 3, 7, and 9. Finally, the pathologies. Microscopic ganglionic failure, whether via adult autoimmune destruction in achalasia or infant developmental absence in Hirschsprung's disease, universally prevents smooth muscle relaxation, causing massive, dangerous, and potentially fatal functional obstructions. Thank you for studying with MedStudious. If this deep dive helped you, please subscribe, leave a like, and keep studying hard.